Hello and namaste. Today I came with another new topic. In today's backquotes topic I am going to discuss and demonstrate about the hanging drop preparation. You might be curious why this method is known as hanging drop preparation. This method is called hanging drop preparation because the drop of bacterial suspension which hangs downwards from the inner surface of the cover slips over the concave depression of concavity glass slide. As you can see in the given picture. Let's talk about the application of hanging drop preparation in laboratory. There are the several applications of hanging drop preparation of which it is most commonly used to demonstrate the motility of bacteria. Similarly, it is also used in presumptive identification of pathogen and diseases. For example, hanging drop preparation of diarrheagenic stool. If we see the presence of motile bacteria with darting motility, it suggests that the patient is having a diarrhea because of Vibrio cholera infection and person is having cholera diseases. Now let's discuss about the distinct material required for performing the hanging drop preparations. Most commonly required materials are, firstly we required bacterial broth culture approximately 12-hour-old. Secondly the concavity slide, thirdly cover slip, fourthly Vaseline or grease, petroleum jelly, last but not least is microscope. Now I am going to demonstrate the procedure of hanging drop preparation. First we need to take a clean cover slip and place it into working table. After that, take a wooden cotton stick and apply petroleum jelly on each corner of the cover slip. As we have applied petroleum jelly on each corner of cover slip with the help of inoculating loop. Now we have to place a small drop of bacterial cell suspension on the center of the cover slip. After that, now we need to take a clean concavity slide. Now invert the concavity slide and place a concavity with the concave well facing down, in such a way that the drop of bacterial suspension comes exactly on the center of concavity. Vaseline on the corner of the cover slip which helps firmly to adhere the cover slip to the concavity glass slide. Now slowly turn or flip the concavity slide so the cover slip is on top, on which you can see the drop of bacterial cell suspension seen hanging down from the inner surface of cover slip on the concavity well of concavity slide. Now place the concavity slide on the microscope and focus the drop of bacterial cell suspension first at lower power, 10x, and locate the edge of the drop. Once you had located the edges of drop, then turn the objective to high power objective, 40x. Focus on the edge of the drop carefully and look at each side of that line for very small rod or spherical shaped object that are the bacteria. If bacteria is motile, you will see moving. Let's see the example of motile bacteria. Mnemonics for the motile bacteria is she purchased vest. Motile bacteria are Salmonella typhi and Salmonella paratyphi, Helicobacter pylori, Escherichia coli, Pseudomonas aeruginosa, Proteus species, Vibrio cholera, Enterobacter species, Spirillum minus, Treponema pallidum,